Next up is Richard Gaughan. His is Seeing Hidden Structures with Holographic Microscopy. Is that right? It is. Oh, thank you. See, I'm a musician. I'm really impressed with myself right now. He's pursuing his Master's of Science in Applied Physics, and Christopher Mann is his major advisor.
Let's say you've got a friend from North Carolina. Your friend comes out for a visit. You go to the Grand Canyon, Wapatki, out to Painted Desert. Your friend thanks you for being a great tour guide and then heads home. 6 months later, you learn your friend is in the hospital, suffering from valley fever, made worse because it wasn't diagnosed for months after the symptoms first appeared. Here at NAU, in a tiny basement laboratory, we're working on a method to lay the groundwork to test and identify those difficult to diagnose diseases. Our method depends upon a principle of physics that allows us to see transparent microscopic objects. And this is important because these diseases, although they present with very non-specific signs, like fever and rash, they have very specific molecular signatures. All right, if there's very specific molecular signatures, why doesn't that clinician in North Carolina just draw a blood sample and look at it? Because what they'd see is something like those blood cell images you see from the CDC right there. Biological cells are essentially transparent. You can't see anything. It's like looking at a bunch of pieces of glass in a glass bowl filled with water. Now, current imaging techniques rely on special sample preparation to add color to those microscopic structures, as if each one of those shards of glass was given a different color. The problem is those sample preparation techniques are expensive and time-consuming. And no clinician is going to order an expensive and time-consuming test for a rare condition until they eliminate those options that are more likely. And in North Carolina, valley fever is not very likely. Now, our method replaces that special sample preparation with a special imaging technique that requires no sample preparation at all. It's called digital holographic microscopy. And whereas in a regular microscope, light just goes through a slide and you take a look at it, in digital holographic microscopy, we take a laser beam and split it in two parts. One part goes through the sample and the other goes around. When the two are brought back together, they create a pattern of darkness and light. And we mathematically process that pattern of darkness and light to reconstruct an image of those hidden structures within the cell, like you can see on the image on the left with the, oh, you're right, sorry, with the structures shown inside these blood cells. Now, right now, we've already proven the principle. We're working on adding a new capability, combining many images together to produce a 3D model of these cells. For now, it's a research capability, but eventually, it could be the foundation of a method to quickly, accurately, and inexpensively detect difficult to diagnose diseases. Thank you.